Hello. Meeting. World. Meeting കഴിഞ്ഞിട്ടോ? Hello, sir.
Hello, good afternoon.
Good afternoon, sir.
Good afternoon to all. It gives me immense pleasure to welcome each one of you to today's webinar organized by Kerala Biotechnology Commission and Kerala State Council for Science and Technology in observance of World Hepatitis Day 2026. On behalf of KBC, I extended a warm welcome to our Honorable Member Secretary, Dr. P. Harinarayanan, to the session. And I welcome our distinguished speaker, Dr. Agiesh Kumar, sir, Senior Principal Scientist, Institute of Advanced Virology. We are grateful to him for accepting our invitation and for sharing his expertise on timely and significant topic, biomarker for viral disease, labs to clinic. Welcome, sir. I welcome Dr. Sharika A.R., Principal Scientist, KBC, KSCSTE for this webinar. I also welcome all participants who have joined us today. I cordially invite Dr. P. Harinarayanan, sir, Member Secretary, KSCSTE, for introductory remarks.
Good afternoon. The speaker of the day, Dr. Agiesh Kumar, BP, my colleagues, Dr. Sharika, Dr. Sumaya, and my dear participants. As we all know that we are celebrating the World Hepatitis Day today to commemorate the birth anniversary of Dr. Baruch Samuel Bruberg, the Nobel laureate and the scientist who discovered the hepatitis B virus and developed the first hepatitis B vaccine. This year's theme actually is hepatitis. Let us break it down. It's a call to simplify the conservation around hepatitis by raising awareness, dispelling myths, reducing stigma, and encouraging timely testing and treatment. It reminds us that knowledge is one of our strongest tools in combating this silent disease. Viral hepatitis remains a major global health challenge. Millions of people are living with hepatitis B and hepatitis C, often without knowing they are infected. As the disease may remain asymptomatic for many years. If left undiagnosed and untreated, it can lead to serious complications such as liver cirrhosis, liver failure, and liver cancer. However, the encouraging reality is that hepatitis can be prevented, detected early, and in many cases, effectively treated or cured. Recognizing the importance of these advancements, the Kerala Biotechnology Commission, under the Kerala State Council for Science, Technology and Environment, has organized today's webinar on biomarkers for viral disease, the lab to clinics. These sessions aim to bring together researchers, clinicians, healthcare professionals, faculty members, and students, and out to the public to gain insight into the latest developments in biomarker research and the translation into clinical practice. I congratulate the team once again and open the workshop. Thank you very much.
Shall I start?
Sir, before starting the talk, I give a small overview about our distinguished speaker, Dr. Agiesh sir. He had his PhD in biotechnology from Pondicherry Central University. He completed his postdoctoral studies from Texas Tech University and University of Georgia, USA. He authored 60 plus international publications and he is serving as an editor and associate editor in many reputed journals. He is a recipient of prestigious ICMR DHR International Award for Senior Biomedical Scientist and he is a visiting fellow at the National University of Singapore. His research mainly focused on the endothelial protein and the role in understanding the chemical outcomes of viral disease, and his work aims to identify predictive biomarker and to develop prognostic device to improve disease management in infectious disease. A warm welcome to Dr. Agiesh sir to the session, sir.
Thank you, Dr. Sumya. And at the outset, I thank KCST and Dr. Harinarayanan and my colleague Dr. Sarika for the kind invitation to uh, give a talk on commemoration of the World Hepatitis Day. So I shall be sharing my screen now.
Yes, sir.
So is it visible?
Yes.
Yeah. So the topic which I'm going to discuss now is on biomarkers for viral diseases, that is from lab to clinics. And in my presentation, the first part of my presentation, I thought I'll devote some time on, giving an overview of hepatitis virus and the biomarker that is, I mean, what are biomarkers and how biomarker discovery is being done, and some of the biomarkers that are currently available in clinics. And I will also talk about the biomarkers for hepatitis, the current scenario. Then the later part of my presentation, I will also briefly touch upon about my research on biomarkers and the kind of collaborations I'm currently having. So this is actually the genome architecture of RNA viruses, and I thought I will give an overview of how viruses are being classified. And I have brought in only the RNA viruses because most of my research is on RNA viruses, and the major virus which is there is hepatitis B virus, which comes under DNA viruses, which is not being shown here. And under RNA viruses, we have a single strand RNA virus and double strand RNA virus. And in a single strand RNA virus, if you look at some of the important viruses like envelope, I mean, again, it is classified into envelope and non-enveloped viruses, whereas all negative strand RNA viruses are enveloped viruses. And again, non-enveloped viruses, we have the hepatitis A virus, that is, which comes under picornavirus. And under enveloped virus, we have the major coronavirus, flavivirus, and alpha virus. And under flavivirus, we have the dengue virus, which is my focus of interest. And also, we have yellow fever virus, JEV, West Nile virus, and hepatitis C virus. So this just to give an idea about how viruses are being classified. And in negative strand RNA viruses, we have some of the dangerous virus like Nipah virus, measles virus, mumps virus, and under arena virus we have Lassa. These are some of the viruses which we have actually this we have reviewed and we have published in current research in virological sciences in 2021. Slide is not. Is the slide moving? Slide moving. Yes. So the slide is getting moved, right? Hello?
Yes, sir, it is moving, sir.
Yes, yes. Okay. So this is actually, like a hepatitis virus, the different types of hepatitis virus, and as we all know, hepatitis it is just the inflammation of the liver, and it is majorly by viral infections, and sometimes it also due to some, some chemicals. And this hepatitis virus is broadly classified by 5 viruses, that is HAV, B, C, D, E, and E. And if you look at the various classifications, and they are not evolutionary, they are not related, or they have been classified mainly under hepatitis because they all infect liver. And again, you have HBV, that is the most severe one. Next comes HCV. Apart from that, we also have HAV, HDV, and HEV. And among all these viruses, only the HBV comes under DNA viruses, whereas all other viruses are classified under RNA viruses. And the transmission pattern is shown here. HAV gets transmitted through fecal oral contamination, and HBV through sexual or perinatal, and HCV again through infected exposure to infected blood, and HDV again, which is infected blood and body fluids, and HVV is again fecal oral, also through zoonotics. And what are the prophylaxis available currently for HAV? We have inactivated vaccine, and HBV again, as we all know, we have an recombinant vaccine, whereas currently there are no vaccines available for HCV, and HDV. HBV can give a protection against HDV as well. So, I mean, most of these are self-limited. HIV, even HDV, all are self-limited, whereas HBV and HCV, they leads to chronic infectious. Yeah, this is again, the epidemiology, or how the virus spreads across the globe. For hepatitis B virus, which has been published by WHO, is shown here. As you see here, I mean, almost across the globe, HPV is being infected, and India is in the top 10 countries for both hepatitis B and C deaths, with hepatitis B tops with 69 percentage of deaths from 10 countries, and hepatitis C comes next to B, that is including 58 percentage of the death. And 1.8 million new infections are coming across annually, and 287 million people are living with chronic hepatitis globally. So what is the message here is integrated molecular biomarker panels holds great promise in the management of viral hepatitis, enabling precision diagnosis, prognosis, and personalized therapy. Sorry, there is some technical glitches. Try move on. Dr. Sumaya, I'll stop sharing and share it
Yes,
again.
yes, sir.
Sorry for the inconvenience. And next slide is on why early prognostic markers are needed of the RR for viral hepatitis. I'll be discussing about what are biomarkers, how biomarker discovery is being done. But before that, we should also understand why the early prognostic markers are needed in the case of viral hepatitis. And like the deaths are increasing despite of the available vaccines or the prophylactic treatments. Now, viral hepatitis related deaths are rising as you could see from 2000 to 2022, there was a sharpened increase in the case of mortality with respect to hepatitis B. And the major concern here is no, most people don't know that they are infected. So the majority of the people with chronic viral hepatitis are being underdiagnosed. And as you could see here, only 10 to 30 percentage are diagnosed, and 70 to 90 percentage remains undiagnosed. So this, like the chronic infection, what happens? It apparently leads to fibrosis, and it left untreated, that get manifest into a liver cirrhosis, and that leads to cellular carcinoma and death. So this is, like this is because of the silent infection, and it also leads to because of late diagnosis, and which results in an advanced liver disease. So what is the need of the RS? Like early prognostic biomarkers for early detection, also for risk stratification. I mean, what I mean, even at the stage of chronic or even acute stage, whether how it can lead to disease severity. And then, like a personalized management, a tailored therapy, and monitor response more effectively in terms of precision medicine. So all these things remains one of the inevitable, or one of the, I mean, the inevitable things for the disease management. Slide movement. In the more system, why it is getting stuck always? Either maybe this can be used. See now it is not getting moved. Next one. That's what I'm doing. Sorry, there is some issue in slide changes. So now I'll move on to the topic, like what are biomarkers and how biomarker discovery is being done. So as we all know, the biomarker is a molecular, cellular or biochemical change that can be accurately and repeatedly measured, and can be used to identify and monitor physiological and pathogenic responses to pharmacological interventions. So actually, like a biomarker is something which helps us to understand the disease process. And here you have various types. The biomarkers are classified into various types. First, let me give a brief overview on how biomarkers are being classified based on clinical applications. So under clinical applications, we have diagnostic biomarkers. That is, that will tell us if the individual is infected or not. For example, hepatitis B virus. Hepatitis B virus, now it can be easily diagnosed based on the surface antigen. So the biomarkers which will give an idea of the presence or absence of infections. Next is prognostic markers. Prognostic markers, as the name indicates, that will tell us the without any intervention what would be the outcome of the disease. Like whether that individual may develop into severe form of not at an early point of time. So that is prognostic biomarkers. On the other hand, predictive biomarkers. Predictive biomarkers is something, with intervention, what would be the outcome. So with by giving an intervention, how the whether the disease progresses, or the disease prevention I mean the disease virulence has been reduced or not. And next is pharmacodynamic response markers. The drug which we have given had given any intended effect or undesired effect. That will tell us pharmacodynamic markers. monitoring biomarkers, something which longitudinally, if you want to monitor the patients after giving a drug. For example, our insulin level can be easily monitored based on the level of HbA1c. So based similarly, safety biomarkers. So these are some of the biomarkers that is being classified based on clinical applications. On the other hand, this is how clinically how the biomarkers have been classified. But now, as a researcher, we also would be deriving informations based on our research. For example, genomic biomarkers, anything, the changes can be predicted based on the levels of DNA or proteomic biomarkers at protein level. Similarly, RNA, that is transcriptomic biomarkers, that is any changes that happens at RNA level, and that can be used as a biomarker. And now lipidomic biomarkers, which is currently in an emerging, like that will based on the levels of lipids, we can actually we will be able to understand the disease prognosis or disease diagnosis. Epigenetic markers, wherein it is actually not there is no change in the DNA per se, but at the epigenetic level. Then some of the cellular markers. So these are some of the markers that are being currently developed based on research, and many markers are being developed for the diagnosis of both viral and metabolic diseases. And moving on to the imaging and physiological functions, physiological biomarkers like blood based on blood pressure monitoring, imaging biomarkers based on imaging and EEG, ECG, all those comes under imaging biomarkers. And based on all these biomarkers, and again they're classified based on the disease, something for the detection of cancer, that is oncology biomarkers, cardiac biomarkers, troponin is one biomarker that will the clinicians are able to understand the development of any cardiovascular diseases. Those comes under predictive biomarkers. Neurological biomarkers, infectious disease biomarkers, inflammatory markers, CRP, the change in the levels of, based on the levels of CRP, we would be able to understand what is the infection, status of infection. So all these can be classified under inflammatory markers. So this slide essentially gives us an idea about what are biomarkers and what are the types of biomarkers. Yeah. Then, like what is the current state of the art? I mean, how biomarker discovery is being done. So before getting into that, like we should understand, we should also get an idea about what are the characteristic features of an ideal biomarker. So what is an ideal biomarker? So if you ask me or anybody would be happy if the biomarker is non-invasive, like without any prick, or if you don't, not through device, directly if you are able to measure. For example, SpO2 is being measured with, it's a non-invasive made and easily measured. The biomarkers, whatever technology we are going to develop, that is going to be, I mean, it should be easily measured, and apparently it should be inexpensive and also produce rapid results. I don't want anything to wait for one week or 3 weeks, 3 days. By then the disease would actually disappear. So like, or it will also manifest into in a severe form of the disease. So like it should produce a rapid results, and it should be available from a readily available sources. And two important character in which a biomarker should have, there should be a balance between two properties. One is sensitivity and the other is specificity. So when I say sensitivity, that is, it should be highly sensitive. That means it should be allowing early detection, and there should not be any overlap in values between patients and controls. So to put it simple, highly sensitive means the biomarker should identify a patient, I mean, whoever is infected as infected. That means it should not give any false negative results, and it should give whoever is infected should accurately predict or precisely predict the patient with somebody with the individual with the disease. On the other hand, high specificity. Here it is being greatly upregulated specifically in the disease sample. So high specificity means it should identify a control as a control, that is negative as a negative. So there should be a proper balance between sensitivity and specificity. And biomarker should also, like vary rapidly in response to treatment, And it should also aid in risk stratification and possess prognostic value in terms of real outcomes. That is, the biomarker would be able to help us in understanding the outcome of the disease. And above all, so these are some of the properties. And as a researcher, I'm also interested to understand the mechanism of the disease. Like if the biomarker in just instead of playing a role of just a mere biomarker, it should also give us an idea about how the disease mechanism takes place. So it should provide some insight into underlying disease mechanism. So these are some of the features of an ideal biomarker. And what are the sources from which a biomarker can be? It can be from the whole blood, serum, plasma, saliva, urine, CSF, peritoneal fluid, pleural fluid, tissues, etc. And what are the approaches that are being followed? Like I have developed a biomarker in the lab, but how I convert that biomarker in the form of a point of care device, that is important. That should be served to the public. Like lateral flow. Now the lateral flow devices are being assays are being developed, and ELISA kits are being developed. PCR based assays, like PCR based assays were highly helpful in term in detecting the virus, especially during COVID time. Then HPLC, FACS, mass spec, multiplex protein microarray. These are some of the sophisticated techniques that also will help us to identify a biomarker. So once I have, supposing if I want to develop a biomarker, what are the different stages in which a biomarker discovery should undergo? That is from discovery, translation, and validation. So if you look at this, there are 5 different phases. From preclinical discovery, that is wherein like discover biomarkers in tissues or body fluids, confirm and prioritize promising candidates. So supposing if I have identified something, if I speculate that these are some of the markers that could actually help for the detection or prognosis of a disease, then actually I should start with the preclinical discovery. Then I should actually develop an assay. Like for example, if I want to detect some marker, I can develop an assay. For example, ELISA or PCR, whichever lab-based assays should be developed. So once I have preclinical assay development, then I should actually do a retrospective analysis. Like whatever the results which were already available. So test biomarker in a completed clinical trial. Test if biomarker detects the disease early. Evaluate sensitivity, specificity, and ROC. So that is the proof of concept one should develop before actually going for a prospective screening. So if I have to submit a project or something on biomarker discovery, I should have actually undergone all these things done beforehand, and then actually I have to go for a prospective screening. Use biomarker to screen a large population. In my later slide, I will show how actually we have done from a preclinical to prospective screening for in our study. And then once you have done all these things, finally the biomarker actually should have given an impact, how by controlling the disease, whether the biomarker has given the intended effect by reducing the disease burden. So all these 5 stages the biomarker should go through to make the biomarker an effective biomarker. Yeah, so this is, like this one. So now that we have discussed about what are biomarkers and how biomarker discovery is being done. So now to give an overview, I mean, what are the current biomarkers that are already available in clinics? Irrespective of, like viral or any, irrespective of hepatitis or any other viral diseases, what are the biomarkers that are currently being used in clinics? As we all know, the CBC, whole blood count, will give us an idea about whether there is some infection, there is some alteration, even like for example, if the lymphocyte count goes down, platelet count goes down, that gives an clue that there is some viral infection. Similarly, NK cell count. On the other hand, some of the biomarkers like AST, ALT, these are 2 important biomarkers that will tell us if there is something happening in liver, if there is some alterations in liver or liver damage or something. ALT, AST is the best markers that gives an first-hand information that there is something happening in the liver. And similarly, bilirubin, blood urea nitrogen, LDH, creatinine, and albumin level. All these will help us in understanding the viral infections. And coagulation markers, D-dimer. D-dimer is one biomarker which is highly used to understand the coagulation status. Even during COVID, patients were advised to check D-dimer once a while to understand the coagulation status. So these are some of the markers that, apart from bacterial viral, it also helps us to understand the metabolic diseases. And CRP, as I mentioned, it will tell us any inflammatory response, and like if there is some infection based on the CRP levels. I mean, clinicians were able to understand what is the status of infection. But though there are several biomarkers are available, but the concern is not all these biomarkers fulfills the criteria which I have mentioned in my previous slide. That is, like it has to have a high specificity and high sensitivity. So I have mentioned that there should be a proper balance between sensitivity and specificity, but apparently these biomarkers does not fulfill that, but still it gives an idea. Though there is a limited sensitivity and specificity, it gives an idea that the kind of infection that takes place. So again, back to the topic on hepatitis, I thought I will give you an overview of how what are the important biomarkers that are currently used in clinics for the diagnosis of hepatitis, especially towards precision medicine. For example, some of the markers here, that HBV DNA and covalently circular DNA and quantitative surface antigen, and CR, that is correlated to antigen. So all these antigens are will helps us for the diagnosis, and not only diagnosis, it will also give an idea of viral replication and treatment eligibility, like based on the amount of antibodies, and monitoring antiviral response, and all these things are possible based on the HPV whatever markers. And the ALT, AST, and CK18, these markers will give us an idea of the liver injury and necrosis and disease activity. All these can be obtained based on ALT, AST. And some of the other markers, like I have also mentioned in the classification, there are some genomic markers, there are some proteomic markers, metabolic markers. Like that, they have derived some of the other markers that will actually tell us what is the staging of fibrosis, whether from chronic towards fibrosis, and who are at risk for cirrhosis. All those things, the main application is it alleviates or reduces the need for liver biopsy. Then the important biomarkers, IL-6, some of these, they may not be highly specific, but still it gives an, after immunotherapy, what is the level of immune activation, exhaustion, and disease progressions can be understood based on the levels of inflammatory markers. And some of the other HBS AG and HBS-CR-AG, it helps for the prediction of cirrhosis and HCC for long term, and it also will help the clinicians to understand what is the long term clinical outcomes. So, and what are the emerging biomarker landscapes? Like what are the emerging or current state of the art technologies that are being explored for biomarker discovery? Apart from the techniques which I have mentioned in my previous leads, starts from lateral flow assay till flow. Some of the other methods that are currently being employed includes digital PCR, liquid biopsy. That is, that helps for, it actually reduces the need for liver biopsy, and multi-omics techniques, transcriptomics, proteomics, metabolomics, whatever I have mentioned before, it actually helps us to understand the stages also for a precision medicine in hepatology. And AI integrated panels are also being developed for disease stratification. And WHO's priorities towards 2030, that is strengthen prevention of mother to child HBV transmission, scale up antiviral treatment, integrated hepatitis services into primary healthcare, and expand universal screening. So all these things are priorities made by WHO. with reference to hepatitis management. So, with this, I will now I have given you an overview of what are biomarkers, types of biomarkers, how biomarker discovery is being done. Then, what are the biomarkers that are currently used in clinics? What are their, advantages and limitations? And what are the types of biomarkers that are being developed or already employed in clinics for hepatitis management? Now, with this, I will move on to the kind of research which I am doing on biomarker discovery. And the first area which I'm focusing on is bacterial viral differentiation. Most of the case times the bacterial and viral infections produce with similar clinical symptoms, and it makes it challenging for the clinicians to distinguish between bacterial and viral infections. And what happens, this leads to inappropriate usage of antibiotics, and this actually leads to the spread of antimicrobial resistance. And India has the highest antimicrobial resistant mortality rate in the world. And addressing this challenge is crucial in optimizing antimicrobial stewardship and mitigating AMR crisis. But what is the thing is, currently there is no single perfect reference standard for diagnosing bacterial and viral infection. And traditionally available methods such as bacterial cultures, PCR based pathogen detection, and even the serology methods, it has its own limitations in terms of sensitivity, specificity, and turnaround time. And as we all know, culturing bacteria also slow, and even sometimes it may fail to detect intracellular or uncultivable pathogens. So what is the alternative mechanism which we are exploring now is, instead of directly and targeting the pathogens, we are targeting the host response. And, as we all know, the bacterial and viral infections are expected to target different signaling pathways. In the case of viral infections, it is expected to target the interferons or cytokine related pathways, and this leads to the production of many proteins like CD46 trial MXA. On the other hand, bacterial infections targets the integrin related proteins, and this leads to a different transcriptomic signature, and this is expected to produce a different set of cytokines or proteins. So targeting these proteins, we expect that could be able to differentiate between bacterial and viral infections. And what we did a biocomputational study. We did a meta-analysis, and in which we have, sorry, we have taken around 48 data sets, including 4,637 samples across 19 countries. And we had three sets. One is discovery set, and discovery set had six data sets. And from this, we had confirmed bacterial infections, confirmed viral infections, and confirmed control samples. And whatever data we have derived from the discovery set was first validated with a validation set, which included 10 data sets. And whatever we filtered through validation set 1, we later confirmed with validation set 2. And apparently finally we ended up with 7 bacterial gene, and 7 viral and 9 bacterial. So this is the overall flow from bacterial core response 540 and viral core response 309 we have derived. And we did a differential analysis. We filtered, we narrowed down to 30 viral and 70 bacterial, and from there we confined to 7 viral and 9 bacterial. And what is the key advantage? What we presume is the collective data analysis. We presume this is robust and reliable biomarker identification, consistency across multiple studies, and it also minimize the influence of study specific bias. And this, we feel it improves the data integration by collective normalization of data from different studies. And this also enhances detection of subtle changes in gene expression patterns via increased statistical power. And we have done a bioinformatics study, and we have also have done a pilot study. Pilots. In my first slide, I have said biomarker discovery steps involves a preclinical phase. From then, wherein you have to go for a retrospective and then do a prospective. So that is what we did based on the bioinformatics retrospective study. We actually did with a small set of patient samples, and we have found some of these markers works pretty much good in differentiating between bacterial and viral infections. And now that what we are doing is we are currently taken up a project which is supported by ICMR, and in this we are validating a host RNA marker panel to diagnose bacterial and viral etiology in undifferentiated acute febrile infections, and this we are doing in tie-up with Believers Church Medical College and Hospital. So here, the aim is to assess the diagnostic performance of the host RNA biomarkers identified through a robust cross-platform data integration approach in prospective patient samples, which I have mentioned in my previous slide. And we also, once we have the data from the prospective patient samples, we also would like to develop a machine learning based classification model for the accurate differentiation of bacterial and viral infection. And interestingly, this is the largest set of samples, unlike the previous pilot data. Here we have five groups. One is confirmed bacterial cases, confirmed viral cases, and indeterminate cases, wherever the lab data fail to identify properly. So where there is no certainty, and we had one set of healthy controls. We also have incorporated a non-infectious inflammatory and autoimmune diseases that would actually help us to say whether the whatever biomarkers we develop, which actually it's highly sensitive and specific in terms of detecting viral infections. So this is the sample size calculation, and this is the inclusion and exclusion criteria which we are currently doing. I think that is not much needed. And what is the expected outcome? So the study outcome, what we are aiming is, we are based on successful completion of the study, we would be able to develop a reliable panel of novel host RNA markers differentiating bacterial and viral infections. And by then we will have a clinical validation of identified biomarkers in a cohort of confirmed patient samples will be done. And also we would have developed a cross-platform data integration pipeline for biomarker discovery. And we also would have developed ML-based classifier with superior sensitivity and specificity. And also we will have a data set of patient samples with confirmed bacterial, viral, and indeterminate cases for further studies. So here I would like to mention that the confirmed viral cases, we have both all viral cases, including hepatitis B and C cases we have in our data sets, and we are also including other viral infections like dengue and SARS-CoV and influenza. So this is one study which from the biomarker thing. The first study is about biomarker discovery for the early differentiation of bacterial and viral illness. The next project which we are involved is the dengue biomarkers for dengue. So in the case of dengue, the major concern is how some people who develop into severity is not known. I mean, who may develop a severe form of the disease at an early point of time when the patient comes into the hospital, and once when they are confirmed with dengue viral infections, after a period of time, most of the patients will be fine, but some patients may develop into severe form of the disease. But currently there are no methods available to say who may develop a severe form of dengue. So the type of study which we are involved, I mean, which we have taken up now is to develop a cost-effective point-of-care prognostic system for debilitating viral disease like dengue and SARS-CoV. So this is just a background. Patients come. I mean, they undergo one after the mosquito bite. Once the virus gets transmitted, and they develop into a febrile phase of disease, and then they develop into critical phase, and then finally recovery phase. But this critical phase, that as I mentioned, most of the patients should be fine after the febrile phase, that is 4 or 5th day of the 6th day of infection. But after that, they may develop into severe form of the disease. So what is the urgent need is to clinical need for identification and validation of potent biomarkers for early prediction of severe dengue. So what we are doing here is we are targeting some of the endothelial proteins, like dengue virus are known to infect endothelial cells. So we are targeting some of the normally what happens, the endothelial cells are intact and it maintains a vascular integrity. But whereas during dengue viral infections in severe forms, what happens? It hampers the endothelial integrity, and this leads to dengue severity. And we are targeting some of the proteins. So the proteins released by activated endothelial cells are expected to be a better predictor for severe dengue and disease outcome. So this is what we are targeting. And like we have characterized some of the proteins that are released by endothelial cells like endocrines, cytokines, MMP14, PCAM, and we found that some of these proteins are exclusively upregulated in severe form of dengue, and currently we are interested in knowing whether these proteins can be a better predictor for severe dengue. And like as we all know, in the case of dengue, the platelet counts goes down, and hematocrit plasma leakage is there. And so we have found that these proteins are directly associated with the platelet count and plasma leakage. And we wanted to know whether these proteins could be a better predictor. And we have made a collaboration with the University of Singapore, and wherein we did a machine learning model, and we understood that some of these markers could predict the dengue with higher precision. And we also we are interested whether they are just biomarkers. In the first slide I said, are they just mere biomarkers, or they have anything to do beyond that? I mean, anything to do with the disease pathogenesis. That is what my team is looking at, whether some of these markers have anything to do with clinical manifestation. This is just one mechanism which we have proposed based on our research findings. And we have also found that some of these proteins are found to be associated with oxidative stress markers like protein carbonylation, DNA damage, and protein carbonylation, DNA damage, and lipid peroxidation. And the results we have published in Free Radical Biology and Medicine. This is one protein which we have taken up now, that is guanylate binding protein. And this protein is very interesting because these proteins are known to have antiviral infections, antiviral activity. And now some of the papers, they say even for hepatitis, these proteins could be an effective target for developing antiviral therapy. So because they are antioxidant proteins, maintaining the levels of these guanylate binding proteins will help the cells to overcome the infection. Once during viral infections, the levels of these proteins are expected to go up. But what happens when as the disease manifests or disease progresses, it tries to suppress the levels of these proteins because of the oxidative stress. That is what I have shown in my previous slide. There is a correlation between viral infection and oxidative stress. So as the oxidative stress goes up, we have found that some of these proteins are going down. So the idea is to, if we maintain the level of these proteins, antioxidant therapy can actually augment the levels of these GBPs, and whether it can actually helps for the cells to overcome the oxidative stress. So that is what one concept which we are working now. And as I mentioned, the other protein among guanylate binding proteins, we have so far characterized three. One is 1, 4, and 5. So 1, 2, and 5. So this GBB5 is already known to have an it is known to have an antiviral activity in the case of HBV. And now that we are working with reference to dengue virus to see whether it actually modulates the antiviral response, and it helps for the patient to recover by maintaining the endothelial integrity. So that is what we have made the hypothesis, and we have published this in the Journal of Medical Virology. And here we have seen once the severity increases, there is it leads to hampering vascular integrity and leads to endothelial dysfunction. On the other hand, when the patient recovers, the endothelial integrity is intact. That is by maintaining the levels of GBP. So now that we have taken up some in vitro studies, maybe in some of my next presentations, I will have something to show how they have a role in maintaining the homeostasis in the case of dengue and HBV. So these are some of the markers which we have identified using metabolomics. So in my first slide, I have said proteomics, metabolomics, genomics, right? So some of the metabolomic markers which we have identified during the severe form of viral infection is shown here. This again we have done in collaboration with the Indian Institute of Chemical Technology. And the results were published in Metabolomics. So what is that we have done now is, I mean, so far whatever I have said, it is based on the pilot study. So now that we have taken up a large scale study wherein we want to understand the dynamic changes of these proteins in severe cases, and whether these markers will help us to understand the disease prognosis. That is what we are trying to do, integrating in vitro and machine learning models. And in this study, we have, this is the overall aim of the study. Previously, we have followed up the patients till the recovery phase. Now that we want to develop a marker which will help the patients, will help the clinicians to understand the disease outcome at an early point of time. So we are focusing only the patients who comes in the hospital with the febrile infections, and we want to screen those patients, and we want to see if our markers could be a better predictive markers for the understanding the outcome of the disease. So here, so based on our pilot study, now we have taken up a prospective study which involves around 2,275 patients, which has both severe and non-severe categories, severe form of dengue and non-severe categories. And here we are collaborating with three hospitals, Believers Church Medical College, Pushpagiri Medical College, and Trivandrum Medical College. And we have started getting samples, and we are in the screening. We have started screening the markers which I have explained before. No, we have started screening how the markers behave in a multicentric population. And same, like endothelial dysfunction markers. So some of these markers, as I mentioned, they work well for dengue, and some markers even helps for hepatitis. Though I have not directly worked on hepatitis, there are papers that says that it helps for understanding the prognosis of hepatitis and hepatitis B. And we, since SARS-CoV also involves endothelial dysfunction, we wanted to know whether these markers could predict the help for the understanding the COVID disease outcome, COVID-19 outcome, and some of these markers worked pretty much well. And these results we have published in some of the papers like Biochemy, IUBM Life. And what is that we are currently based on the lab data, and we have done some machine models, and now that we are tying up with both NUS and other industry people in developing a device. So wherein we want to develop a device two in one. I mean, both the diagnosis, also the prognosis. It will also identify who has a viral infection, also who may develop into severe form of disease using prognostic biomarkers. So that is what we are trying to develop. And not only viral infections, we also, I mean, this is actually in a preliminary stage now. We also want to know how this viral infections actually apparently, I mean, biomarkers helps for a viral associated heart failure thing. That is one thing which we want to work on, and we have made a review on this on in heart failure reviews. And the idea is we know that obesity, CKD, COPD, diabetes, hypertension, microplastics, which is a currently added one, and hyperlipidemia, all these things leads to endothelial dysfunction. But what is how the virus can actually, influence or regulates this endothelial dysfunction. So some of the basic knowledge which we have generated so far, we want to, extrapolate, or we want to scale it up on how they actually targets, I mean, how it has anything to do with CVD also. So that is one thing which we are trying to. We are in initial talks with the Sri Chitra Medical College on this line, and we may come up with a project on this line. So these are some of our papers which we have published on viral biomarkers so far. And we have also edited a book on viral hemorrhagic fevers, which recently got published by Elsevier. And I would like to thank my collaborators. As I mentioned before, I have active collaboration with the Believers Church Medical College and Hospitals, Mahatma Gandhi Medical College and Research Institute Pondicherry, Trivandrum Medical College, and Pushpagiri Medical College. And we have also, for mission models, we have a collaboration with Institute of Chemical Technology, National University of Singapore. Now that we have started collaborating with our bioinformatics division at IAV, and we get funding. We are largely supported by funding from DBG, CSAR, ICMR, and SERB, and other things. And this is my team. The left one is my previous people from my previous employment. My PhD student Vignesh, who actually had done major part of this endothelial markers. And Sanjita, she is the one who's worked on the GBP. Ashika, she worked on lipidomic stuff. Salman, Rajesh, they have worked on endothelial markers again. And the right one is my current people. Dr. Shanas, who actually helped me in making the slides, some of the slides, and she is working on this endothelial marker, the in vitro stuff, whatever I have mentioned. Then Dr. Rajesh, he is working on bacterial viral discriminating markers. He is also helping us in, developing machine models for the data which we have generated, and Lakshmi is helping us in doing all molecular assays. And before I wind up, I also would like to say, as an associate editor of IJMR, so this is a prestigious journal of ICMR, and it publishes high quality research. And if now their timelines are it's within 5 to 7 weeks, we will be able to hear the first decision, that is after review. So if we have any data, interesting data, you may be think or consider in submitting to IJMR. And this is the recent coverage which I thought I will share. Like in the last Sunday Hindu edition, there was a coverage on our biomarker studies on viral diseases has been published. Thank you, and I'll be happy to take up any questions.
Thank you, sir.
Mm.
Wonderful and very informative talk, sir.
Thank you very much. I think some in between there was some, problem in moving the slides.
Oh, it's okay, sir. It's fine.
Sorry about it.
Okay, sir. Dear students, now the session is open for discussion. Dear students who have any questions, please ask. And the participant kindly fill the feedback form to get the certificates also. Sir, one question is in the chat box, sir.
Yeah.
Sir, can I read, sir?
Yeah, please, no?
Sir, given that endothelial dysfunction is a common feature across multiple hemorrhagic viral infection, can single endothelial markers reliability predict severity for specific viruses always like in the case of dengue, or will we always require multi-marker signature?
Yeah, actually, this one, now that we have many markers, but apparently when it comes to sensitivity and specificity, it is not highly good enough for diagnosis or prognosis. So if it is a multi-marker, it will be really good. Like a combination of markers would give a better result than a single marker. So that is what the machine model does. So what is the best combination? What is the best fit of markers that could give a better predictive efficacy? So that is what I feel is multi-marker rather single one. Because sometimes most of the times the clinical manifestations would be the same. So in that case, if we have a markers, 2 3 markers, that will work well fine than a single marker. Yeah, that is taken in clinical trials in India. So, clinical trials in India per se, with reference to hepatitis, I think I have, like the one which I have shared in my slide, some of the markers, those markers I mean, per se in India, if hepatitis biomarkers, to my knowledge, which is again under clinical trial, I don't think so, but I may have to check before I give you a proper answer. But some of the biomarkers that are available in clinics, that has been shared already. But what the biomarkers, some this GBB is also one of the biomarkers, since it is involved in endothelial dysfunction, that is again well studied in hepatitis. But clinical trial, I'm not sure that is being done actually.
Thank you, sir. The session is live on YouTube also. You can see some comments came from the miscom due to network issues. Like the session is live on YouTube and all. You can see it. The other question is came, sir. Why do some biomarker work well in research but fail once tested on real patients?
No, it is like, that is what when it should be we should how the study design was done. Like if you want to if you test only with a few patients, it may work well, and other confounding factors also will be there. We will not know. We may think that the results are because of the biomarkers, but when we actually do with a proper case control, and we should have a age and sex matched control. We should also try in multicentric samples. So just by doing in 10 or 20 samples, we may get good results. That doesn't mean that that's a good biomarker. Like we actually have to work on all the confounding factors, because confounding factors also will help, I mean, will add on to your results. So that is why sometimes what we see in the lab may not happen when we take up to in a large scale.
There is one question, sir, from Reshmi R. Nair. Given the recent developments in the dengue vaccine research, what biomarkers are considered most promising for predicting vaccine efficiency and long-term immunity in the Indian population?
No, there actually, I just saw this morning. I mean, like they have taken up, they have mentioned it gives 80 to 90% sensitivity, other details they have not mentioned, I think. What are the biomarkers that actually, but if for any immunogenicity neutralizing activity, based on IgM, IgG, for any viral diseases, neutralization activity only they'll see whether it can actually suppress the a virus. I mean, neutralization property. Any good virus should have a good neutralization property. So that is how they would have done. They would have done also for vaccine per se. There actually again biomarker, when you say viral load should be minimized. Viral load should come down. Also, it also in the case of dengue, if you talk that ADE, that response secondary, irrespective of primary or secondary response, it should not lead to disease severity. So those mostly on viral numbers, viral copy numbers they might have done. So that is taken as a marker here.
Next question is from Kale Selvi. Many biomarkers are common in different viral infections. How do you choose the best biomarker for a specific virus?
Yeah, that's a good question. Actually, like when we go for biomarkers, it is like, the response is the same. So, how do we say, for example, when I say, how do I confident I am, how confident I am that it is exclusively for dengue? That is not possible. But, like I can compare with other febrile illness. For example, scrub typhus. Scrub typhus and dengue, mostly it will have the same, symptoms. So in that case, if some of my markers are exclusively higher up or down in dengue, then it shows that those markers are highly specific towards dengue rather scrub typhus. So I should also have a control apart from your healthy volunteers. I should also have a control wherein they are negative for dengue, but they should also have other febrile illness properties. So that is one in my cohort. I will also have other febrile illness as one of the controls to rule out the non-specificity.
One other question from Shruti. Sir, could you please brief on the guidance for analytical and clinical validation of biomarkers and the number of samples required?
The number of samples required varies from individual experiments to individual experiments. For example, in one of my studies, my sample size is 425. That is based on the prevalence of the disease. Prevalence of the disease in, I mean, cases that reports to the hospitals. Wherever I'm collecting samples, I have to make a sample size based on the number of cases that report to that particular hospital. On the other hand, the other study, my sample size is 2,225. That means I have multiple hospitals, and again based on the number of diseases that report to the hospital. So sample size differs. And again, supposing if I have a marker, I also should have publications or previous studies to understand the expression of that marker. So based on the expression of the marker, I have to recalculate the sample size. So sample size calculation is not the same for all the experiments. It varies.
I think the questions are over. Watch the video on the session on YouTube. The link is already shared in the chat box. And then I am inviting Dr. Kriba, consultant of TBT Skill Vigyan, to share the vote of thanks.
A very good afternoon.
One more question.
Excuse me. One more question is there from Sangeeta. Although HCV is successfully cleared following DAA therapy, some patients continue to progress to cirrhosis or hepatocellular carcinoma. How can biomarkers help identify patients at risk of disease progression despite viral clearance?
Yeah, despite viral clearance, actually, this is one common thing with reference to viral diseases. Like irrespective of even in dengue also it happens. Though there is no virus in circulation, patient develop disease severity. That is because of the individual immune response and the inflammatory response that takes place in an individual. So that is how, like the precision biomarkers I have shown. Some biomarkers are exclusively helps for tailor-made or individual personalized medicine. So whatever we say, it may not be completely ubiquitous. We say it is a generalized terms, statistically we determine, but it may not be the same for everybody. In some of the cases, their host response also determines. I mean, it will have a major role. So it varies actually.
There are no other questions. So a very good afternoon to one and all. As we come to the close of today's insightful webinar organized by the Kerala Biotechnology Commission, KSCSTE, on the occasion of the World Hepatitis Day 2026, it is my privilege to propose the vote of thanks. First and foremost, I express my heartfelt gratitude to our distinguished speaker, Dr. Agiesh Kumar BP, Senior Principal Scientist, Clinical Virology, Institute of Advanced Virology, Trivandrum, for delivering such an informative and engaging talk on biomarkers for viral diseases, labs to clinics. He has given an overview on the types of biomarkers, current status of the biomarker research and biomarker discovery pipeline, and also your expert insights into the importance of prognostic molecular biomarkers for the early detection and personalized management of viral and bacterial diseases. It has greatly enriched our understanding of the rapidly evolving field. We sincerely appreciate the time, effort, and enthusiasm you devoted to interacting with the participants and patiently addressing their questions. Thank you, sir. We are sincerely grateful to Dr. P. Harinarayanan, Member Secretary, KSCSTE, for his inspiring inaugural remarks, which aptly underscored the history of the behind the observance of Hepatitis Day, the significance of scientific awareness, early diagnosis, and the collaborative efforts needed in addressing this viral hepatitis. His continued guidance and encouragements have been instrumental in the successful organization of this program. I also sincerely thank all the participants, students, faculty, scientists, researchers, and students for their active participation and interest in this webinar. I extend our sincere thanks to Kerala Biotechnology Commission for organizing this webinar as part of the World Hepatitis Day observation. Such initiatives provide an excellent platform for connecting researchers, clinicians, students, and healthcare professionals with leading experts and for promoting scientific awareness on the issue of public health importance. I also take this opportunity to specifically thank the officials and staff of KSCSTE, especially Dr. Charika Iyer, Principal Scientist, and Dr. Sumaya Ashraf, Junior Scientist, the technical support team, and everyone who worked behind the scenes to ensure the smooth planning and seamless execution of this webinar. Once again, we express our sincere gratitude to Dr. Agiesh Kumar for sharing his valuable expertise with us. We hope today's session was inspirational to all the participants, and it would inspire you to further explore the exciting role of biomarkers in advancing viral disease diagnosis and patient care. Thank you all for your presence and active participation. We wish you all a wonderful day ahead. Thank you.
Thank you, one and all. Thank you, KCST, for the opportunity. Thank you.
Thank you, sir.